Let us look at ocean acidification and metals. So ocean acidification alone or with the pollutants can affect many aspects of physiology of marine organisms, including acid-base balance, energy metabolism, redox balance, as well as behavior. The metals in the marine environment originate from both the anthropogenic and natural sources. But for metals such as cadmium, lead, zinc, nickel, copper, the anthropogenic emissions significantly exceed the natural input. So this is what is the thing we have to keep in mind. And of course, the changes in the seawater chemistry due to the ocean acidification can affect solubility, speciation, and distribution of metals in water and sediments. This affects the metal toxicity to marine organisms. So reduction in pH increases the solubility of metal. For example, nickel, iron, cadmium, zinc, lead, and copper. All these things get dissolved in water more under acidic conditions. Ocean acidification will result in the elevated level of most bioavailable free ionic form of metals which coordinate with carbonate anions. For example, Cu2 plus ion, Fe2 plus, nickel 2 plus. Whereas the metals that coordinate with the chloride ion are not affected due to the change of ocean acidification. Because when we talk about ocean acidification, we are mainly focusing on carbon dioxide gas getting dissolved in water. So that's the reason it leads to the formation of carbonate anion, and any metal which can coordinate with carbonate anion will be affected, whereas any metal that can coordinate with chloride anion is not affected that much. So the bioavailability of metal uptake and accumulation is more complex process. It's not a straightforward chemical reaction which we can simply write. And that's the reason it is very difficult to predict using chemical models, because chemical reactions within the flask are happening very effectively. We know what is the individual species which are involved, but when it is biological systems, it is a much more complex network of things that are happening. The availability of free metals in the seawater does not always translate into metal accumulation and toxicity of the species. So this is also another thing we have to keep in mind. So it basically depends on the life stage of the organisms, the degree of acidification, metal contaminated sediments. So there are various factors which are involved in that whole process. There is a Pitzer interaction model which can be used to explain that. So this model is used to understand the effect of pH on metal speciation. So the stability constant beta for the formation of complexes in pure water is used for understanding the metal ion interactions or complexation in seawater. So what we are doing is we are simply doing a comparison between pure water and the seawater. So for hydroxide, the formation of complexes are expressed as a stepwise hydrolysis of metals. A metal in the N plus ion form reacts with the I molecules of water to give hydroxide anion, hydroxide product. So this is the simple reaction. So the metal N plus ion is getting converted to metal hydroxides, and which releases H plus ions. So the change of ions are actually, the charge of ions are actually given here. When we write the hydrolysis constant, beta I is given by this particular equation. Since the ratio of the total carbonate to free carbonate is constant at a constant salinity and temperature, the formation constant for carbonate complexation can easily be expressed in terms of total ion concentration by the following equation. So this is what the Pitzer interaction model, and the limitation of this model is this only considers the formation of strong complexes and it doesn't consider the formation of weak complexes with chloride and sulfates. So that is the major limitation. Here are the table that explains about this particular process. So both hydroxide ion and carbonate form strong complexes in ocean water with metals that are divalent and trivalent. So here you can see this color, this purple colored ions are basically Al3 plus, it's a trivalent, Cu2 plus, it's a divalent. So either it may be a, or beryllium 2 plus is a divalent, nickel 2 plus is a divalent, gallium is a trivalent. So of course thorium 4 plus and rare earths are also there, but The inorganic ligands, basically the hydroxides and the carbonates, form complexes with the divalent and trivalent anions. These metals will undergo significant change in the speciation as the pH of the seawater decreases. But metals that form strong complexes with chloride or which are mainly in the free form will not be strongly influenced by the change of pH. So this is the major difference. So for example, silver, gold, copper, cadmium, mercury, all these form strong complexes with chloride, or there are some free or weak complexes like Mn2 plus, In2 plus, CO2 plus. These are not affected by the change of pH. And of course, there are certain metals which doesn't go any change, like for transition or the mixed inorganic ligands like Pb2 plus, Y3 plus. These metal complexes does not undergo any change. And of course, there are other metal complexes which are given here also does not undergo any change. The ocean acidification and metals, when we continue, the metals aluminium, gallium, indium and beryllium, these form very strong complexes with hydroxide and are not hydrolyzed by a pH change. But there may be a speciation change. That means Al3 plus is getting converted to Al4 plus species, or AlOH3. So that means the change of speciation actually takes place when the pH changes, but these are not getting hydrolyzed. Similarly, metals that form strong complexes with carbonate, they also does not undergo changes. The rare earths will be the most strongly affected by the change of pH, which results in the increase in their free ionic form. So these species can form complex with chloride and sulfates. So that is what actually the change happens to these particular metals. Lead and yttrium are in the transition or the mixed group. So lead forms chlorides at lower pH. And yttrium, the transition from carbonate to chloride ion also occurs when the pH reduces. Similarly, for Fe3 plus, Al3 plus and As3 plus, they are highly soluble in both acidic and basic medium with low solubility in between. So depending on the change of pH, the solubility may increase or decrease because the difference is very, very small only. When the pH changes, either it is highly acidic or highly basic, then only the change happens. See, unlike the inorganic complexes, there are metal organic complexes of iron, cobalt, copper, zinc and lead that are major species found in the ocean. And photosynthetic organisms basically require manganese, iron, nickel, cobalt, copper, zinc and cadmium. But the high concentration of copper, lead and cadmium are toxic in the marine environment, because we will be studying in the later part that marine pollution. So at that time we will be mainly studying about some of these metals. Iron 2 plus, nickel 2 plus and lead 2 plus, all these 2 plus ions form strong complexes with organic ligands in seawater. So far we saw about the inorganic complex. Now we are looking at the organic complexes. And similarly, the inorganic ligands are pH dependent. So measuring them will give information regarding inorganic form. So that is what we saw, because we have already seen carbonate can exist as a bicarbonate anion also. But it is very difficult to identify whether these forms are beneficial biologically or toxic is very difficult. So that is the major limitation. When the metal organic complexes are strong, they cannot interact with and affect organisms. So because they don't actually get assimilated by the plants or animals. So that is the reason the biological system cannot use any strong metal complex that is already formed outside of their system. So some of the organic functional groups that form complexes with metal include phenolic and carboxylic moieties. These are pH dependent, because we know acidic compounds, carboxylic acid, can exist in two different forms. Under highly acidic conditions, it will remain as basically in the acid form. But under higher, alkaline conditions, they may exist in the carboxylate anion form. Similarly, the phenol also can exist as a phenoxide ion. So these are all the two species. They change when the pH changes. The limitation is the non-homogeneous composition and unknown structures of the organic ligand makes it very difficult to identify and characterize which organic ligand is capable of forming metal complexes in the marine environment. So this is the major drawback or the challenge we have. The metabolic rate, ventilation rate and the assimilation rate of food of aquatic organisms directly influence the uptake of waterborne metals. So this is also another thing we have to keep in mind, because a species exists in the marine environment does not and need not necessarily mean that it will be consumed or it may be used by the biological animals or plants that exist in the seawater.